Hi, I'm Carissa Lau from MCDB 427, Molecular Biology at the University of Michigan, and this video is about evidence for a ternary complex involving HDAC2, Mad1, and Sin3A from experiments in the cell paper. Histone deacetylases associated with the Sin3 co-repressor mediate Mad transcriptional repression. Let's review some important background information. So there are immense amounts of DNA in a cell, located in the nucleus, and not all of it is transcribed. There are many histones in the cell, but we're just going to draw a few to illustrate concepts. Anyway, to organize DNA, the cell wraps it around proteins called histones, like wrapping your excess laptop charger wire around the block to form a complex called chromatin. Now, chromatin is very important for a process called gene silencing. If an area of DNA is lightly wrapped, RNA polymerase can't reach it to transcribe genes, but if it's unwrapped, RNAP has enough room to bind and begin transcription. These different states of chromatin are called euchromatin, which is active and loosely wrapped, and heterochromatin, inactive because it's so tightly coiled, silencing the gene by blocking transcription. In other words, one reason gene silencing occurs is because DNA is in the heterochromatin state. So then, how does DNA change from euchromatin to heterochromatin? One answer is acetylation. In euchromatin, DNA is acetylated, meaning that acetyl groups are bound to the histone tails, allowing for euchromatin to be loosely wound around the histones themselves. When euchromatin is deacetylated, a protein called a deacetylase removes the acetyl groups, allowing DNA to wind more tightly around the histones. Gene regulation also occurs through repression, where proteins called repressors bind to specific regions in DNA. Repressors also have helpers called co-repressors, who bind to them and indirectly regulate gene expression. These processes are important for the cell to control when and how much a gene is expressed. Now let's get to our actual experiment. At this point in time, we know our proteins of interest repress genes through repressor-directed deacetylation mediated by a co-repressor. But now I'll show you how the authors get there. First, our proteins. We have HDAC2, our histone deacetylase, Mad1, a transcription factor that binds to DNA, and Sin3A, a mammalian co-repressor which ties the two together. The authors wanted to examine how these proteins associate or otherwise work together, doing so through an experiment which I'll recreate and walk you through. Throughout the data, the goal is to ask and answer, what is the interaction between these proteins? First, here's some of the cells tested. This experiment used a human embryonic cell line. Each cell has some combination of two different types of plasmids. The first type of plasmid contains only a gene for either flag, a small peptide epitope, by itself, or flag HDAC2, which is HDAC2 tagged with flag for ease of immunoprecipitation. Anti-flag antibodies will bind to the products of either plasmid. The second type contains either no Mad1 gene, a functional copy of Mad1, or Mad1 Pro, which is a mutant copy of Mad with a proline substitution that was previously shown to prevent binding to Sin3A. Let's look at what happens when you lyse some of these cells without any immunoprecipitation, which gives a whole cell lysate with all proteins in the cell.
Separate the proteins on SDS page and detect on a Western blot using antibodies to Sin3A and Mad1 to visualize the proteins, since no proteins will be visible until an antibody probe binds to them. Looking at lanes 7 through 9, we have the plasmids in each lane in the header on top.
Each lane has a plasmid expressing flag protein and a plasmid expressing either the plasmid vector alone, Mad1, or Mad1 Pro. Using antibodies to probe for Sin3A and Mad1 in these lanes shows that the cells express Sin3A regardless of the plasmids they received, that the plasmid transfections of Mad1 and Mad1 Pro were successful, and that the Mad1 antibody binds to Mad1 Pro. Overall, the cell lysate lanes show that Mad1, Mad1 Pro, and Sin3A are all expressed where we want them to be. Now we want to look at how our proteins associate. One way is to use an immunoprecipitation column, IP for short. First, pour your solution of interest through a column of beads, and the antibodies embedded in the beads bind their epitope from the solution. By comparing IPs for the flag and flag HDAC plasmids, we can easily control for association to HDAC2 by comparing which proteins precipitate out. These comparisons are also how we know flag HDAC is expressed where we want it to be. By using anti-flag immunoprecipitation and antibody probing, we can narrow down how Mad1, Sin3A, and HDAC2 interact. Basically, the IP only pulls down proteins associated with the target epitope, so using this data, we can show which proteins interact with which others. In lanes 1 to 3, we IP from cells that have a plasmid containing only flag and a plasmid containing either no Mad1, Mad1, or Mad1 Pro. IP with an anti-flag antibody shouldn't pull out any HDAC-associated proteins. Probing with anti-Sin3A and anti-Mad1 shows no visible complexes. So since we know from lanes 7 through 9 that Sin3A and Mad1 are expressed, we can conclude that neither of them will bind to the affinity column or the flag epitope.
For lanes 4 to 6, let's change it up and use a flag HDAC2 plasmid instead of just flag. And again, one of either no Mad1, Mad1, or Mad1 Pro. IP with anti-flag to pull down any proteins associated with the flag HDAC2 fusion protein. You can see via an anti-Sin3A probe that Sin3A precipitates in each of these lanes now that we've added HDAC2, meaning HDAC2 and Sin3A are associated in some way. Let's probe for Mad1. When we don't express Mad1, we don't see it immunoprecipitate, which is what we expect, and no band appears as shown in lane 4. If we express a wild-type Mad1, it does immunoprecipitate, and we see this in lane 5 since a band appears. In contrast, when they express the mutant called Mad1 Pro, which we are told does not interact with Sin3A, it does not precipitate. This is shown in lane 6, which does not have a band. Remember that Mad1 Pro's mutation removes its ability to bind to Sin3A? Since Mad1 Pro doesn't IP out, Mad1's ability to bind Sin3A is necessary for it to come out in a complex with HDAC2. That implies Mad1 binds to Sin3A, which binds to HDAC2. To remind you, we know from the cell lysates shown in lanes 7 through 9 that Mad1 and Mad1 Pro were definitely expressed by their plasmids. So let's review the overall implications of this figure. Because Sin3A immunoprecipitates when flag HDAC2 is expressed, but not when flag by itself is expressed, we know that Sin3A binds directly or indirectly to HDAC2. We can see this by comparing lanes 1 to 3 and lanes 4 to 6. Mad1 also immunoprecipitates when flag HDAC2 is expressed, but not when flag is expressed by itself. So we know that Mad1 also binds directly or indirectly to HDAC2. However, we have Mad1 Pro, the mutant we are told cannot bind to Sin3A, and since Mad1 Pro does not immunoprecipitate with flag HDAC2, we know it does not bind to flag HDAC2 directly. These experiments overall show that HDAC2, Sin3A, and Mad1 form a ternary complex, with the potential order being Mad1 to Sin3A to HDAC2.
Thanks for watching, and go blue!